right
In
around.
progress.
I want to thank Dr. Brower for an excellent M&M, and I want to thank you guys for excellent answers at M&M. Thank you for your participation. So today we're very pleased to have the Wangensteen lectureship. I'll start with a few comments on that. Dr. Wangensteen from here, your Dr. Wangensteen, is widely regarded as the architect of modern academic surgery. And I still believe today this is the case. His most transformative contribution was the creation of a training paradigm that integrated rigorous laboratory science with clinical care, establishing the modern surgical scientist model. He built one of the first research-intensive surgical residencies, really emphasizing, if not mandating, dedicated investigative training to producing an extraordinary lineage of leaders, including everybody that you see in the hallways around you, and I dare say all of you someday. And I mean that sincerely. His clinical innovations, particularly in GI physiology and the use of NG decompression for bowel obstruction, reshape perioperative care. Just think about that for a minute. How many nasogastric tubes people get? There was a time where we just didn't know to do that, and think about the mortality of bowel obstruction at that time. He reshaped perioperative care through these contributions, and he transformed Minnesota, this department, into a global center of surgical innovation and redefined surgical education. In that same tradition, today's Wangensteen speaker, Sylvester Black, exemplifies a contemporary surgeon scientist. And Sylvester used to sit right over there by Sean Wynn. I remember it so well. It's a long time ago. Truly exemplifies this. As a graduate of our program here and the PhD program, you completed an advanced fellowship training in abdominal transplantation and hepatobiliary surgery at Toronto, University of Toronto, and currently you are the surgical director of liver transplant at Ohio State, the Ohio State University. You lead a very high volume training program, clinical program, alongside a very robust translational research enterprise. Your most significant contributions center on advancing organ preservation and machine perfusion technologies, and you've been almost consistently supported by the NIH, Department of Defense. You currently serve as the chair of the NIH Hepatobiliary Pancreatic Study Section, helping to shape the national research agenda in transplantation science. You published a lot, over 75 papers at your young stage of your career, at least. Your career, and here's something I will never get, his career has been recognized by multiple national honors, including the Presidential Early Career Award for Scientists and Engineers. It's incredible. And the highest honor bestowed by the U.S. government on an early career scientist, as well as the American Society for Transplant Surgeons Rising Star Award. You've been repeatedly named the top doctor for your scientific and clinical achievement, and I can't think of a better person to be the Wangensteen lecturer. So I'm truly honored, we're all honored that you would come and present today. Thank you, Dr. Black.
Wow, it's really an honor to be here to see a lot of my my faculty members that trained me, residents that I did training with, and just a lot of friends, and to be here in this room, a lot of memories were here. I remember where I used to sit, and kind of Dr. Chipman's questions were very familiar. In terms of the tone and temperament, but it was great. I definitely miss this place, and it's really a huge homecoming and an incredible honor that Dr. Crombin asked me to come back and speak. Now, these are my disclosures. Today, it's going to be a little bit of a potpourri on the field of transplantation, a little bit about myself, a lot about Minnesota, and just really where I think the field is heading. The field is, I think, in a transformative period. This period is probably as transformative as the period that ushered in transplantation with immunosuppression, where we're going to proceed to precision medicine. And so it's really incredible. Now, thank you, Dr. Crombin, for giving this slide for me. I can't really say much more about Owen Wangensteen other than that he really shaped the paradigm of what it means to be an academic surgeon. And I can't say that enough. And that when you go to the surgical meetings and you look at the nasogastric tube, the surgical forum, the Society of University Surgeons, and the whole concept of a surgeon scientist, he exemplified that. And that drew me to Minnesota. Minnesota was the center of gravity in academic surgery. And you can go to any meeting and there's leaders that either had close contact with people at Minnesota or were from Minnesota. And so in transplantation, especially in cardiothoracic surgery and surgical oncology. Now, my journey began in 2001 as a intrepid young surgical intern. And it was an incredible experience. Things were a little bit different at that point in time, but I met people from the University of Minnesota in my medical school at Northwestern, Joe Leventhal, John Fryer, Dixon Kaufman. There were a number of people, and they said you had to go to Minnesota. If you wanted to be a surgeon scientist, that's where you had to be. And so I went here with that in mind, and really was a really transformative experience for me, and in a lot of ways shapes my worldview on academic surgery. And I was able to get my PhD in the laboratory under Gus Del Maso, and that was back in the days before autocorrect. And Gus wasn't too great at typing, and so I sat there. He was writing R01s, and I basically was his autocorrect. And he had many times thought that English wasn't my native language, and we struggled through many R01 submissions, and I was able to get an RSA as well. And I learned how to be a surgeon scientist. And for many of the faculty members here, I also learned how to be what it is to be an academic surgeon. And so it really brought it together and really kind of codified how I view the world and really set me up for success. And when you look at transplantation, this is one of the cradles of transplantation here at the University of Minnesota, but transplantation was often thought of in ancient times. There was a lot of mythology around transplantation, where you can take and recombine animal parts and people in various ways. And you look at the Chimera of Arezzo, or the patron saints Cosmas and Damian exchanging a soldier and a parishioner's leg. You look at Prometheus and the whole idea of liver regeneration and losing your liver and having it regenerate the next day. We've always kind of had an idea that transplant involved, that it was certainly technically feasible and possible. And so really transplantation, excited me as a young surgeon scientist from what I saw with Dr. Najarian and the transplant center here, with the fusion of science and the imagination and the clinical acumen of the surgeons there. And it was really just a fantastic experience. And so that kind of led me to transplant and then also the history. But let's look at transplant itself and look at the history of transplantation. Now, transplantation, I'll pick this one point as one of the foundational parts of transplant history. And this is President Sadi Carnot, who in 1894 was stabbed in the abdomen, and it went right through his liver and into his portal vein. And so the surgeons of the day had no ability to repair vascular injuries. So they struggled trying to put suture in and ligature, and the patient died on the table, exsanguinated right on the table. And there was a very interesting individual here who was the foundation of kind of a lot of modern surgical practice, very controversial individual, and often can be seen as really the kind of first, I don't want to call him the father, but one of the first progenitors of a transplant, a cardiothoracic, and a vascular surgeon. Now, Alexis Carrel, he has a very difficult kind of social history, and I don't want to get into that aspect of it because a lot of his views were certainly unpalatable at the very least. But he was a genius scientist, and kind of some of his contributions we still do today. We still teach the residents, and he really set the field up in showing us what is technically possible. Really brilliant person. If you think about things like cell culture, if you think about things like vascular suture, if you think about things like organ transplantation, the heart-lung machine, normothermic machine perfusion, that's all Alexis Carrel. Now, Alexis Carrel was a trainee in the operating room when Sadi Carnot was stabbed in the abdomen, and he was absolutely horrified that we could not fix this. And so he went and got in touch with a seamstress in France, and he learned how to do minute vascular repair on vessels. And then he went and actually formed, the modern concept of what the anastomosis is. He took fine silk suture, Vaseline and was able to do all these very intricate repairs, and he showed that it's feasible. Then he went on to do organ transplants. So he would take hearts from dogs and put them in necks. He would exchange limbs of animals, and they would do well initially, and they were well vascularized. But they ended up dying, and the organs ended up not functioning over time because he ran into what he described as a biologic force that could not be overcome. We know that today is the immune system. But he kind of gave rise to the idea that it's technically and surgically possible to replace body parts. And so when you look at the history of surgical transplantation, I want to look at it in three eras. The first era is the technical feasibility. Can we do the operation? And so Alexis Carrel started that, and he showed that it was technically possible to do the nuts and bolts of the operation, do the vascular anastomosis, to replace organs. That certainly was feasible. Then in 1933, Yuri Voneroy actually took a kidney from a deceased patient and put it into the thigh of a patient in renal failure, and the kidney functioned for about 6 hours, and it was making urine. The patient ultimately ended up dying, but he showed that it could be technically possibly done in a human. And then in 1954, Joseph Murray, who went on to win the Nobel Prize, did a kidney transplant with the Herrick twins, and they were identical twins. So he took the immune system out, thought like a true scientist, and was able to win the Nobel Prize, but took the immune system out of it as a variable and showed that it was technically feasible. And they lived 8 years. And then he went on to do a lot of work in non-identical twins to show that it is in fact technically feasible as well. And then in the 1950s to the 1960s, we were able to start doing kidney transplants. Everybody got the prodigious amounts of steroids, but John Najarian, Folkhart Belzer, and others were able to show that it was technically feasible, that these were DCDs in the day, so they waited till the donor's heart stopped, they took the kidneys out, they did the transplant as quickly as possible. Usually the donor and the recipient were in close proximity, so it was able to be done, and they showed that it could technically work. Then the 1960s were the golden age of technical transplantation, many of the first which happened here. Richard Lillehei did the first pancreas transplant. Tom Starzl did the first liver transplant. Christian Bernard, who was a fellow here along with Norman Shumway, and they studied in the laboratory together. And Owen Wangensteen gave him the heart and lung machine, and he traveled back to South Africa after learning the technique from Shumway, and actually did the first heart transplant before anybody else. And so that caused a lot of controversy in the field, but that was also in Minnesota. Both of them were in Minnesota. And then James Southern Folkhart Belzer invented University of Wisconsin solution, which allowed us to actually take donor organs and preserve them for longer periods of time to make transplant more technically feasible. Now, the immunosuppression era, we learned about immunosuppression in this second era, whether it was Peter Medawar looking at transplant and immune tolerance and showing us, giving us the modern basis of immunosuppressive drugs. And a lot of his work with skin grafts and mice showed that it was technically feasible, and that the immune system, there was a scientific basis for suppressing the immune system. And then the late 1950s and 60s, Roy Kahn and others worked with antimetabolites and added that to steroids to allow for more effective immunosuppression, things like azathioprine and ultimately mycophenolic acid analogs. John Najarian and the University of Minnesota team shows up again with Minnesota ALG and induction therapy, taking immune globulins and depleting lymphocytes, and then Tom Starzl as well with ATG, or antithymocyte globulin. And then Boreal and others, Najarian, Starzl and Kahn worked with the calcineurin inhibitors, and that kind of ushered in the modern era of immunosuppression, where calcineurin inhibitors were in fact the kind of engine that allowed us to really do transplantation successfully. So what we're going to talk about today, mainly in this talk, is the whole concept of era 3, the precision medicine era, where we're pumping organs, where we're modifying the organs on a genetic level, and we're also practicing precision medicine with advanced therapeutics. We're really kind of getting at the scientific underpinning about, around making organs survive longer and matching them more effectively. Now, nobody can argue with the success of modern organ transplantation across all organs. Survival rates are greater than 90% in almost every organ, and even the 5-year survival rates are great. It's one of really the modern miracles in modern medicine. And so it took a while to get here, but nobody can argue with the success compared to waiting on the wait list. And the mortality is very high across all organ systems if you don't get a transplant. The biggest problem with transplant is we don't have enough donor organs. For the number of patients that get on the list, greater than 100,000, we do about half of those in transplants per year, but that's a really selected patient population. If you look at liver transplant, for instance, of the 6 people that get worked up, 1 eventually gets put on the waiting list, and then a fraction of those get transplanted with a significant waitlist mortality. We don't have enough organs for transplant, so we have significant unmet need in this country and really worldwide. If you look at the number of waitlist additions and the number of people that actually die of end-stage organ disease, it dwarfs it significantly. And so the question that you have to ask yourself is that if you had enough organs, if all the barriers were removed, and that you could do as many organ transplants as you wanted to, how many lives could you impact? Because if you look at end-stage organ failure, it's upwards or close to 1 billion patients. And so if you had an infinite number of organs, and if you could make those organs work, then you should be able to save a significant amount of lives. And so this lack of suitable donor organs is the question. It's not a lack of donor organs, it's a lack of suitable donor organs, and there's a difference. Now, as surgeons, we've thought of all kinds of creative ways to expand the donor pool. We cut livers in half and make split livers and give 1 to a kid and 1 to an adult, or 2 to adult, depending on the size of the adults. We do live donors, where we take part of the liver of a living person or a kidney and give it to another patient. We've expanded what we consider a donor. Back in Starzl's day, everybody was a young trauma patient, 20 to 30 years old. Now, I recently am getting calls from my partners about the 81-year-old DCD that we're going to pump and use. And so we have to kind of contend with all these comorbidities to expand the donor pool. These organs obviously have significant comorbidities and injury, and during the process, you have to figure out how to manage that injury. And that's where precision medicine will come in. And, we talked a little bit about live donation. All the surgeons, we love it. It's a technical tour de force. You get to map the liver and do all these complicated vascular reconstructions. It's a lot of fun from a technical aspect. And, this is liver and kidney as well. But I do consider it a little bit of a failure from the modern societal system, in the sense that we have to take organs from living people, expose them to risk to help somebody else. In a perfect world, we would recover as many of the organs, transplant. See Dr. Adams there, is that we would have an infinite supply of organs, and we wouldn't have to expose our donors to risk. So if you're in Asia, this is the only kind of organ that you're going to get, because there isn't a pool of deceased donors due to culture and other reasons, that it's just not a thing. So living liver donation living donation period is a great thing. But hopefully, if precision medicine keeps advancing, we won't need living donors in the future. This is our program that we started in 2018, and these are the very first 2 people that we did. And it was a great operation, went well. And we have a small, live donor program, mainly because we are so aggressive at using marginal organs that we really haven't had the ability to expand this. But it is useful in many cases when you can't get an organ because of the allocation of the patient and not having high enough criteria scores to get those organs. Now, deceased donor organs are primarily where most of our organs come from. There are 2 types of deceased donor organs. There's donation after brain death, where we wait we have a patient that has cessation or irrecoverable brain function and can't be is declared dead for a variety of reasons and can be used as a donor organ. And donation after cardiac death, which I think this might be the first year, but its DCDs are starting to outpace what's available for donation after brain death. And there's certain implications for that, which we'll talk about in terms of the quality of the donor organ. Now, there are a number of things that have set up transplant that allow us to do transplants in the modern era. And you think about 2 significant legislative things and 2 significant legislative and biological criteria or acts that allow us to do transplantation. The first is the 1968 Harvard Ad Hoc Committee report, which defined what brain death means, and we'll talk about that in a minute. And the next one is the 1981 Uniform Determination of Death Act, which gave a legal framework and declared what is a dead patient, whether it's brain dead or circulatory death, and allows us to actually do transplantation currently. And so that's how you get modern transplantation. And there are a couple of problems that we're going to have to face in the near future with this. And really, I want to focus on the Harvard report of irreversible coma, the idea of irreversible coma, the idea of brain death. As you can imagine, as science pushes further and further, we're able to redefine things that we thought were impossible. So this idea of brain that is dead and what is brain death are being challenged on many levels. And what does this mean ethically? What does this mean medically? If we can recover a liver, why can't we recover a brain? And so these things are starting to kind of show up in transplantation. And there are moves, and you may have seen some of this in social media and in the news, where the organ procurement organizations are kind of having to wrestle with what is a dead donor, what is a DCD, what is a brain dead. And we're starting to, as a community, feel the ramifications of that. And we'll talk about that in a second. And so this idea of brain death, and these are some interesting papers that kind of show how it's being challenged, that the kind of idea of consciousness and the kind of idea of what it means to be you and me and everything, and we've relied on brainwaves, and brainwaves can flatten, but they're looking at, you know, kind of how that happens, and can that be reversed? Can you start to get synchronized brain activity again? And they're starting to study that, and the answer, at least in some of the in vitro work, appears to be that there may be some hope for that to happen. And so then you look at this next article, and this article was really interesting, where they took pigs and they euthanized the pigs, and then they waited a couple hours, and then they hooked the pigs up to ECMO, and they were able to get somewhat coordinated brain and metabolic activity from these pigs after 2 hours of ischemic or no blood flow state. So then the question is that the 5 minutes we're using in the current tests that we're using to declare brain death and to declare irrecoverable neurologic function, are those even valid anymore? And so we're looking at that, and that's a significant question that we're going to have to answer. And there are a number of comments in Nature and other papers looking at this. I found this was an interesting article, that they were taking brain cells, or they were taking stem cells, and they basically differentiated those into nervous tissue, and then made organoids, and then hooked them up to a computer, and they outperformed a large language model. And then now there's some other papers where they've hooked these organoids up to robots, and they're able to control kind of movement in the robots. So there's a lot of interesting things that are happening. And I saw on social media the other day that there's a company that's looking to take human brains, hook them up to circuits, suppress the brain activity, and test kind of drug responses and things on human brains. So there's a lot of stuff that's going on that's going to cause us to really kind of reevaluate what it means to be a donor and what it means to be brain dead. And so the implications are going to be pretty significant in the field. And so they're looking to redefine and update the U.S. Death Act. And so that's going to have downstream consequences for transplant, because the organs that we're going to get are going to be DCDs with very long warm ischemic times, and how we manage those organs is going to be important. And that's where precision medicine is going to come into the fold. So what is a DCD organ? Circulatory arrest for some variable period of time, or withdrawal of life support, circulatory arrest for some variable period of time. Then we can procure those organs and use them for transplant. Now, as it stands, it's generally, there's a no, we withdraw life support, we wait some period of time until their heart stops. Then we have a no-touch time of 5 to 10 minutes in most hospitals. Then we're able to procure the organs, hook the cannulas up, cool the body, and get the organs out. And then there are a number of technologies that we're using to make those organs, in fact, viable. But you can see that the longer that period of time happens and continues, the more damaged those organs are, especially in the older organs, or the organs that are more marginal. We're doing things now. This is normothermic regional perfusion. This is a technical solution to that, where we hook people up to ECMO, we cut off circulation to the brain and the nervous system, and then we reanimate organs, whether it's the heart or the lungs or the liver or the kidneys. And the results are remarkable. And so this period of not just putting them in a bucket of ice, but actually putting them on an ECMO machine actually has significant benefits for organ function. And those organs do really remarkably well. But there are some kind of ethical questions that we're starting to deal with as a field that we have to address. Is that, and there's some questions that society may force us to address about whether this is we should be doing this. And so then we may have to rely on other technologies such as machine perfusion. Typically right now, what we do is cold static storage. I've kind of mentioned it a little bit. We throw organs in ice after perfusion with an osmotic and energy protectant, which keeps the cells in a hypothermic state. We slow their metabolism down at 4 degrees to about 5%. 5% isn't 0%, so the organs continue to degrade with time and cold ischemia. It works for the most part. It really did allow transplant to expand. However, you can assess organs, and organs that are sufficiently damaged present some significant risk of deteriorating further upon implantation. So this works logistically well enough now, but as the field kind of moves to sicker and sicker donors and donors that have more ischemic time, this is going to be insufficient. And so we've done other things. We've tried hypothermic oxygenated perfusion, or what we call HOPE. where and I don't know how this works. We've argued about this at the NIH level. But what we do is we take cold preservation solution and circulate it around at anywhere between 4 to 10 degrees Celsius, and we oxygenate it. Now, if you think about it as an engineer, cold solution shouldn't hold oxygen without an oxygen carrier for any length of time. I mean, it doesn't work, but yet the results are really good. So maybe it's flushing toxins out, maybe it's modifying the endothelium in some way. It's been talked about the highest levels of government, but the results are hard to ignore. It does work pretty well for extending preservation time and allowing the organs to function better after implantation. Now, here is where we get to the platform where I think it's going to make the biggest change in transplantation. This idea of normothermic machine perfusion, taking the organ out of the body, hooking it up to essentially a heart-lung bypass pump, if you want to think about it, where a pump, an oxygenator, heater cooler, sometimes a dialysis circuit that allows the organ to function at near physiology. And so there's a lot of benefits to that. This is not a new concept. So you'll see here in the corner, Alexis Carrel and Charles Lindbergh. Alexis Carrel was the foremost surgeon scientist in the 1930s and was approached by the famous aviator Charles Lindbergh about a problem that he had. And that problem was that he had an aunt that was dying of rheumatic heart disease. And there was no way at the time to fix the valvular disease in the heart. So Lindbergh, who wasn't a trained scientist, did a few courses in college, but never graduated with an advanced degree, came up to Carrel and said, well, why can't we take the organ out, hook it up to a pump, and fix it, and then put it back? And Carrel thought this was absurd. So Lindbergh pulls out a notebook, and he was fantastic. And he designed his own planes, fixed engines. And so he saw this as strictly a mechanical problem. He drew out this very sophisticated pump and then showed Carrel, and they had numerous lunches at the Rockefeller Institute. Carrel had access to some of the best designers at the time. They didn't have a lot of the advanced biomaterials that we have today, but he had glass blowers and people that could get access to filters and the most sophisticated equipment at the time. They designed this pump that you see in the middle, and he was able to keep organs alive outside the body for extended periods of time. A cat thyroid for 18 days, hearts for 6 to 8 hours, kidneys were able to function. And in the 1930s, went on a world tour showing people all of these organs from animals that were on these pumps. Hearts were beating. It was remarkable. Nobody could repeat the work. Not a single person. Every time they tried, it didn't work. Nobody could figure out how it happened. For some reason, this work got lost to history. And then in 1964, they found some of these pumps in a storage cabinet at NASA. And so the NASA engineers and the people there couldn't figure out how to make it work either. So they call Lindbergh out of retirement. Lindbergh comes in, and they are able to, and these were some of the larger pumps, take a chimpanzee heart and put it on the pump and keep it alive for 6 to 8 hours. It was remarkable. Then everyone forgot about it again. And so then in the year 2000s, they look back at some of this work and that, well, now we have biomaterials, we have heart-lung machines, we have pumps, we have very sophisticated understanding of organ physiology. So they recreated the work with modern materials, and lo and behold, you have the modern normothermic pump. The modern normothermic pump is an ICU for an organ. Now it's going to be utilized in all organs. I think that Dr. Adams can correct me if I'm wrong. I think the kidney transmedics pump is coming out this fall, or maybe the winter. We'll see. And we are able to now keep organs alive for extended periods of time on these pumps. And it's basically, it allows us to actually assess it. So you see livers making bile, you see lungs expanding, you see hearts beating in a box. And in fact, this what got me into to go to Ohio State and hook up with one of my collaborators, and you call him a work spouse, whatever you want to call him. He also was a University of Minnesota graduate, Brian Whitson, and we've ran a lab for the last 15 years. Is that I called him up one day, and I was in Toronto, and this group was procuring a set of lungs that had been transplanted a week earlier, and they looked horrible. And I'm looking over the curtain at these lungs, and they're black, and there was a team from Japan that was procuring for Shaf Deshefri, who's one of the premier cardiothoracic surgeons in the world, and was a leader in this technology. And I said, there's no way, and I was procuring the liver time, there's no way you're going to use those lungs. He said, oh, the lungs are going to be perfect. They'll be fine. Just trust us. So I was very curious at the time. They take the lungs out. I take the liver, and I didn't even care. I didn't say I didn't care about what happened to the liver, but I dropped it off to the implanting team, and I followed these lung surgeons into this room. They had domes, had lungs on pumps. People were bronching. They had 2 or 3 of these things. Never seen anything like it. It was like Star Trek. And so I called Brian up, who was at the University of Minnesota finishing up his fellowship. Brian, we got to find somewhere to get together. We've got to do this. We've got to get involved in this. This is the future of transplant. And so kind of the rest was history, but it certainly was really an incredible thing. And so the studies that have happened with this is that this is the Protect trial, and a number of people were on it. I think they were part of the site here at the University of Minnesota, but this was with Transmedics. We were too, and we tested this and found the machines performed better than static cold storage, and this device got FDA approved. This is the Vital trial with the Organox Mitra, where they were able to take 31 livers that everybody discarded around the world. They put them on these pumps, and then they perfused them, and then they transplanted them with a 71% success rate. These were all organs that were discarded. So this kind of fueled the push for normothermic machine perfusion, and led to a lot of the approvals clinically of these devices. And so Pierre Clavian, who many of you well know for the Clavian complication classification and many other things that he published, he was also a Toronto grad, but he over in Zurich had figured out that we need to pump these things for longer. So he developed a pump that included a dialysis setup, and he was able to keep organs alive for a week. And then he transplanted one after 68 hours to show that almost at 3 days that you can keep these organs alive in the pump for long periods of time. This is important when you want to assess an organ, and if you also want to treat an organ or gene edit or modify an organ, you're going to need to be able to do this. And so, if you look at the methods of organ preservation, this is a cost complexity curve. Obviously, normothermic machine perfusion has the greatest complexity, the greatest cost. In fact, the cost of one of these pumps runs on a normothermic machine anywhere from $40,000 for the Organox Mitra device to $120,000 for Transmedics, but they'll deliver it to you right to your doorstep. So is that sustainable over time? No. The prices are going to have to come down. The economics and economy of scale are going to have to work. But it certainly is something that has revitalized transplantation as of late. And so when you compare all the different technologies, the three things on this table that I'd like to focus on are really the organ evaluation piece, because the normothermic pump allows you to actually evaluate an organ, and that's very important to know how the risk applies to the recipient, the cost, and then also the ability to intervene. You can't intervene when something's in the cold as easily because the liver's not behaving physiologically normal. Any medications and therapeutics you give are not going to work as well. And then, the complications. We've had this. I think most people use the pump. When that pump malfunctions or there's something wrong with that pump, you lose the organ. And so if you do that too much, that can be quite an expensive endeavor. And so that's also an issue that we're working. As the pumps get better, this will get better. And as we talked about with Organox, Transmedics has a concierge service, and you call them up, you say you want an organ, they'll send a team out, they'll get it, they'll deliver it right back to you, and for $120,000, and you'll have to do anything else. And so it's allowed a lot of centers who don't do a lot of transplants, don't have the significant person power to do that. It's allowed them to kind of play in the game. So we've been doing more transplants than ever before, and Transmedics will say that they are responsible for a lot of the kind of increased usage in liver transplant. So really the kind of the meat of everything is towards precision transplantation. This is that third era. The pumps are solved. It's given us a platform. How do we intervene on organs? Artificial intelligence, you've all heard about that. It's going to allow us to make really deep insights onto our data. It's going to allow us to look at organs that are on the pump and make decisions that are definitely statistically and mathematically based. And it's really moving things forward pretty quickly. Precision medicine is also different in transplant in terms of how we look at things. Instead of looking at tissue and instead of looking at typical biopsies and things like that, that we're actually moving to this idea of molecular phenotype, that looking at using deep omics data, looking for new biomarkers, understanding the minute physiology of cells on that level. It's allowing us to really make significant advancements in transplantation. And when we talk about omics, we talk about genomics or transcriptomics. We talk about single and spatial transcriptomics and proteomics and metabolomics, and we get detailed resolution of what is metabolically happening inside the cell. And this allows us really to make very targeted therapeutic interventions. And so you can imagine in organ preservation, we can utilize these technologies to understand how an organ is doing on the circuit in real time. We can evaluate tissue sampling, if we're going to look at biopsy specimens for rejection and understand exactly kind of what is happening and what components of the immune system are at play. And we can look at non-invasive markers, for monitoring how an organ is doing over time. So if a kidney's starting to have early allograft dysfunction, we can intervene earlier and adjust the immunosuppression with more resolution. And so, you see this in areas like the Human Immunome Project, where you talk about omics and you talk about artificial intelligence. We're trying to categorize all the different immune interactions to better understand the immune system at a very high resolution, very similar to the Genome Project and the transcriptome and proteome. So they're doing that with the immunome. We're now doing things like changing the blood groups on donor organs. They do a lot of this in Europe, where they're taking organs from more rare blood groups and creating universal donors, which is going to help expand access to transplantation. We're doing things, and this isn't with precision medicine, but we're still doing work with decellularizing organs and then taking stem cells and repopulating and creating organs. I think that is taking place, and it's also we're also having sorry with the technical. Okay. So we're having success with that. Sorry. One of the neatest things that I saw Dr. Finger walk in, and he gave a talk at Ohio State, is the idea of freezing organs and organ banking. That you're doing some great work at the University of Minnesota with that we can basically take an organ, freeze it, put it on the shelf, and collect and bank organs. And so this whole concept will feed into normothermic perfusion and create a very kind of holistic a way in which we can expand access to organ transplantation significantly. So in my laboratory, we're doing a lot of interesting things. I'm not going to go through the nitty-gritty of each slide, but I'm going to kind of talk a little bit more generally about everything. We have a lot of advanced models that we've developed over the years, which have allowed us to be successful. We have a very large microsurgery center with 2 fully trained microsurgeons who can transplant anything from kidneys to heads on mice. That's if you've ever seen that, that's quite the. I hired him instantly when I saw it, but he's an amazing technical surgeon. We do arterialized mouse liver transplants, and we do at least 10 or 12 of those a week. We have done things like looked at antioxidants. This was this protein called DATO, which basically was found in hibernating tree squirrels. Our idea was utilizing this as a metabolic protectant. This was one of those things in science where you go in thinking one thing and your whole hypothesis actually is correct, but it's still a useful intervention. It was a very potent antioxidant, but it was not a metabolic protectant for human tissue. This was an interesting paper that we put in, and this was where we wanted to figure out a new way to evaluate organs on the pump. And I kind of got this idea, and I was talking to Dr. Whitson. I was standing on the scale, those bioimpedance scales, and I was horrified by what I was seeing when I was looking at it. But then I said to him, I was like, well, how does this occur, right? How are you able to measure fat and water content? And so we went in, and we were doing pigs and other large animals at the time. We found a pacemaker, and we took it apart, and we had some very inventive residents and medical students. We took this pacemaker setup apart, and then we actually put these probes on organs, and we're able to measure the impedance. And what we found is that the impedance often changes, and you can see tissue water before you have organ dysfunction that's detectable in the circuit through traditional means. And so we're developing kind of probes that will fit onto these next generation perfusion devices that will allow us to measure impedance. We also realized that blood for long-term perfusion isn't the answer. So we got when I first got to Ohio State, I went to the head of chemical engineering, and we were talking about, and we developed using these HBOCs, and these are hemoglobin oxygen carriers from cows, where we took the hemoglobin out of cow red blood cells, and that's an infinite supply of red blood cells, pretty much. And then we're able to take the hemoglobin and create very large oxygen carriers that didn't extravasate outside of blood cells and could be shelf stable for 2.5 years in normal conditions. And you just pull them off the shelf, add them to a perfusate, and you have a synthetic blood that is compatible with the circuit, that doesn't break down over time, and allows us to kind of fine tune the amount of oxygen delivery to cells. This was an interesting study, an interesting product that we developed, where we found that when you lift weights and you are working out, your muscles tear and rip, and the membranes tear and rip, and there's lots of membrane breaks, and there's a protein that repairs those membranes and seals the cell. And so we were able to create the recombinant human protein from that, and then we were able to give that. We realized that it actually works for other organs. It doesn't just work for the muscle. So it's like a molecular bandage. And here, I'll show you here, we're poking a muscle cell with a microneedle, and we've used GFP expressing cell, and you can see that that patch in that highlighted area seals the hole. And so we were able to do transplants and actually utilize this as a molecular bandage. So this is a really cool study, and it works for all forms of cell membrane injury. And in fact, we were able to get this published in the Journal of Hepatology for acetaminophen toxicity, and we have similar results for liver and lung transplantation. So we're trying to work to develop that as well. And then my favorite, and the thing that's captured my interest, is this idea of this understanding the metabolism of a donor organ. The metabolism often drives the outcome. So to really understand that on a deep level and to find a pathway that we can intervene on that will have make a difference in those cells. And so CD38 is a master regulator of inflammation and metabolism in the cell. And it basically, its job is to catabolize NAD, and NAD is important in many different things for cellular energetics and all those types of things. And so it creates calcium second messengers and controls calcium signaling on the inside of the cell. GlaxoSmithKline, and it exists in two different orientations. It exists in the extracellular orientation. There's no NAD out in the outside of the cell, very little. There's a lot of NAD on the inside of the cell. But everybody studied the extracellular form of this, and they haven't studied the intracellular form. And so surprisingly, GlaxoSmithKline had developed a drug in 2015 that was effective on the enzymatic level, but when they put it into tissues and animals and cells, it didn't work so well. And so outside the cell, it had a very, very strong binding efficiency and inhibitory efficiency, and it blocked. It looked like the best drug on earth. And then GlaxoSmithKline decided to stop working on it. And they didn't tell anybody why. Nobody knew. And so we found this very interesting. And so I was wondering why it wasn't working. And so I went to talk to my colleagues in medicinal chemistry. But first, I had to understand a little bit about CD38 in the cell. And so this is a fluorescence microscopy showing in the liver that hepatocytes and endothelial cells actually demonstrate a lot of this intracellular form. And so the intracellular form is interesting in that when you have to target a protein or something on the inside of a cell, the cell has a lot of defenses to make sure that your drugs don't affect those intracellular proteins. They have efflux proteins. They have ways to degrade things. It's very, very hard to develop therapeutics that hit and target things without having a lot of off-target liabilities and binding issues on other proteins. And so that was the big thing. And that's why we think GlaxoSmithKline stopped developing this drug, is because it didn't work very well against type 3 CD38. In fact, our experiments have shown that. And so this is another study which we took, and this is an image stream where we took all the cells from the liver and the parenchymal cells, non-parenchymal cells, to quantify the intracellular versus the extracellular CD38. And the hepatocytes in general have a lot more intracellular type 3 CD38, which isn't well studied. And so then we wanted to look at human data. And so we took patients that had DCDs versus brain dead donors. We looked at the CD38 activity, how much CD38 they have. So the DCDs have a lot of ischemia, and so they demonstrate a lot of CD38. They have very little NAD because they utilize all their NAD, and that DCDs in general, even when you pump them, especially if they have significant ischemia, have significantly more injury compared to brain dead donors. And so CD38 is upregulated, and the activity is much higher than the brain dead organs. And so we recapitulated this in a mouse model where we clamped the mouse port up, and then we looked at the injury in wild type mice. And so you see a significant and robust injury with ischemia reperfusion injury in these mice. And then when you give them 78C in very high doses, it's very effective. And then when you look at the knockout mouse that doesn't express CD38, it behaves very similarly to 78C in high doses. But 78C in the doses that we're proposing, or the concentrations, isn't clinically effective. You can't give 20 micromolar concentration of 78C in a human. So we have to develop better drugs. And so we looked at neutrophil activity. How does it work with the immune system? It prevents neutrophil activation, prevents neutrophil infiltration as well when you have the knockout or you get 78C. So it's very effective in that regard. So I approached the chief of medicinal chemistry with an idea. And the idea was, let's develop a class of drugs that went after the type 3 topology. And what we did is we initially showed that the type 3 topology, in fact, is the important topology. We blocked it with a covalent inhibitor. So we blocked all the surface CD38. And then we gave 78C. And what we found is just by blocking the external form or the type 2 form of CD38, that we didn't have any protection. But when we gave the 78C, we showed that it was actually hitting an internal target, likely type 3 CD38. And so to really show this, and this was just with simple fluorogenic assays, LDH, and we repeated this with ALT and everything else. We wanted to show this on a more in a more precise way. So we did hydrodynamic tail vein injection. We created a plasmid that we were able to transfect the type CD38 that we wanted to transfect to show that, in fact, that our hypothesis was true. And so what we did is we first transfected to show that we had a transfection efficiency of about 40%. So 40% of the hepatocytes with our technique were able to be transfected effectively. Then we took a control or a buffer. We took wild type CD38. We took a mutant that didn't have a non-functional CD38, or we took an M21 variant which only expressed the type 3 CD38, and we made the groups and we did those injections. And what we see here is that we're able to, in a knockout population that doesn't have CD38, that we're in fact able to reestablish CD38 activity with the wild type. We're able to, and the M21 variant, so how CD38 is working, and then we're able to actually cause those knockout animals who are normally protected against ischemia perfusion, we're able to reverse that protected phenotype in the knockout animal. Then we're able to re-reverse that by giving 78C and the M21. So we know that type 3 is targeted by CD38. So that led to us developing an assay where we could look at what drugs would bind to that internal form. The medicinal chemists developed this, and they basically they took a fluorophore, labeled 78C with that fluorophore. They transfected hex cells that don't normally express CD38 with this fluorophore, and then we would give inhibitors, and whatever knocked those inhibitors off of that internal form of CD38 or the type 3 CD38 would show up on flow cytometry. So we had the baseline of we wanted drugs more effective than 78C. And so this allowed us to do many thousands of experiments against known drugs that exist, against chemotypes and warheads that had demonstrated, at least by in silico and in the computer, that would have an effect on CD38. And we demonstrated a number of compounds that would actually block the type 3 CD38 with much more efficiency than 78C. And in fact, we were able to develop a number of compounds which are now at the U.S. Patent Office. And this one shows significant tenfold greater effect than 78C against type 3 CD38. So now we have the first in-class compound specifically for type CD38, and we have a lot of experiments now that I'm not showing where we see this effect in mouse transplants. And so this is the medicinal chemistry team. It's like NASA over there. They've got robots that will run these things 24 hours a day. It's important when you're a scientific researcher to get those collaborations early. The second you land as a faculty, you need to reach out like an amoeba and start forming collaborations. And so this team is really great. I've been working with him for the last 6 years, and we've developed a lot of novel compounds. And so that culminates in something I call the Avatar Project, or we call the Avatar Project, where we're able to take cores of tissue from donor livers that are going to be transplanted. So we'll do a wedge biopsy, we'll core out a piece of that wedge, we'll slice it very thin, and we'll create something called a precision cut liver slice. Now, the precision cut liver slice is excellent. I think it's better than an organoid and all these things because it maintains all the cellular connections, it maintains the extracellular matrix and all the kind of networking interrelationships of these cells. So it's basically like a mini liver. Then we're able to take the recipient blood cells of the intended transplant. We can put them together, recreate the conditions of the transplant, and then with our novel compounds, see what benefit that they would have in terms of injury, in terms of genes expressed. We run this all through very high throughput omics. We look at proteomics, spatial proteomics, single nuclear RNA-seq, and a bunch of other things to really understand what's happening both with the cells that are interacting and then kind of all the things that they elaborate and release. What this is going to allow us to do is see the effectiveness of any compounds that we develop, and then on top of that also give us biomarkers for injury. And so then we can look at the recipient donor combination that had the clinical transplant, and we can follow them clinically. So we have both a laboratory result with one-to-one with a clinical transplant. So that will allow us to get a lot of mechanistic insights and a lot of really cool insights that are going to allow us to advance the field. Now, xenotransplantation, really briefly, love it. I created this with AI, as you probably can tell. And I show this at all my xenotransplant talks. And so some people are horrified by it, but I think it looks kind of good. So xenotransplant is really cool. I have a place in my heart for xenotransplant because that's what my PhD research at University of Minnesota was on, and complement and endothelial cell biology around xenotransplant. They've had success, at least technically, with the hearts. They've been able to do some really cool things with, kind of gene editing and humanizing pigs. And, to be honest with you, I never thought we'd get to the day where kidneys survive months. And I mean, it's really been a real technical tour de force. They've done this with kidneys. They've kept kidneys alive for, 2 years in pig and monkey combinations. And they've actually had some success for short periods of time in the human to show that it was in fact feasible. And this is both Bob Montgomery, the first the NYU group, and this is the Mass General group that are both doing a lot of this research. And I found this really interesting. In China, they took a brain dead donor, and then they did a xenotransplant into that donor. And the donor stayed alive, I think, in fairly reasonable condition for 10 days. Then they obviously ended the experiment. And so it is certainly feasible. I have questions about the liver. I think I've talked to Dr. Adams about it before. I think the liver makes so many unique proteins of over 300 to 500, and you're going to have to do a lot of gene editing before you can make that work, because there are going to be some molecular incompatibilities. And I do think the pig may be talking to you by the time you get done gene editing, so you have to be thinking about that a little bit. But I do think where xenotransplantation, and I do think for some of the kidney, I think it's going to eventually work. I think we will get it to work. I do think the heart, I think it's eventually going to work on some level. But where I think that we are learning the most from xenotransplantation is the really sophisticated understanding of gene editing and how all this works and how we change something and see what the immune response is. We're learning a lot. And I remember what Fitzbach, who was one of the big xenotransplant researchers, and Gus Masso and Jeff Platt were in their office and they were talking, and Fritzbach said, even if xenotransplant never works, we're going to learn a lot. And I really do think that the understanding of gene editing is going to really make all of transplant, really a lot of medicine, better. And so how does all this work in the end? And these are the final few slides. This concept of an organ assessment and repair center, which I think is a future hub of precision transplantation. This concept was really interesting. So I was sitting around, and we were in Brian Woodson's office, and we had an empty blackboard, and he loves to write things. He loves blackboards and writes everything down. And so we started sketching out the idea of kind of what a transplant organ assessment repair center would look like and how that would interact with the field. And what we think is that there's going to be some degree of regionalization of transplant. I think with organ banking, I think with enough of these centers that you're going to send donor organs to these facilities, they're going to rehabilitate organs. They may freeze and put them on the shelf. And then, it'll allow for very precise matching of donor and recipient combinations. We wrote this paper in 2014 in a throwaway journal, and we wrote a few other kind of commentaries on other papers. And then I got this call, I think in 2020 or 2021, from the British Transplant Society. And they had read it. And one of their transplant surgeons, I want you to speak at the British Transplant Society on this concept. And Peter Friend and a lot of the normothermic people were there, and they were very excited about it. And, I went to that conference, and they're regionalized. They're doing this exact thing over there. Now, they have abilities to do things with their healthcare system to regionalize healthcare that we are still struggling with here in the States. But they are doing this exact concept where they're going to have all the organs go to these organ assessment centers and then go out for transplant. This is happening in the United States to a more limited fashion. Perfusics was accompanied by Chef Geschäftry and Marcelo Sippel. They're doing a bit of this. We have something similar at the Ohio State University. Brian and I kind of started this early on and went to the hospital, and we reclaimed one of the old plastic surgery ORs and it became our perfusion room. So all of our organs go into these rooms, whether it be hypothermically pumped, kidneys, livers, hearts, all that go to this room. We have perfusion technicians, and they run these organs on the circuit, and then we transplant them. So we're kind of showing that in fact can be done. What this looks like at scale is probably something like this. I think eventually we will get into this idea of meeting the organ supply and quality problem with regionalized precision-based care. And I think that will change the field of transplantation. So just, really quick, some acknowledgments. This is my research team. And they're, I couldn't do any of this without them. And it's really just been a phenomenal journey, and hopefully more good things to come. And then, all the people, sitting in the audience, some of the teachers that I've had, and some of the colleagues that I've worked with at this great institution. Dr. Cromin here for inviting me back. It's really been a pleasure. So thank you.
Questions for Dr. Black?
Quick question and then a comment. We're fearless about bringing up unpleasant ideas.
Yeah.
As a trainee in a difficult clinical situation, you're still fearless. I worry when we do these procurements, at least when I did them, the anesthesiologist would tell me, you don't need to use any anesthetic. You don't need to use any pain medicine. This is a brain dead donor. That always left me uncomfortable. And I think your work, we could bring up the ideas of how is that brain functioning? Are the anesthesiologists still maintaining that they don't need any anesthetic for these brain dead donors?
There have been issues at the OPO level with how donors are handled. They have been in the news. I think that there's significant regulation that's coming. I think our kind of concepts of how we procure organs are going to change in the next 3 to 5 years. I do think that we're going to see a lot longer ischemic times as these issues get brought up, and we need to be in a position where we can rescue those organs, because otherwise we're not going to be doing anything but living donor transplants because the organs are too damaged. Now, as far as anesthesiologists, every hospital kind of has its own policy. And so when you go there and you interact with the OPOs, you know this well. They all have their own little scripts that they go by. And for DCDs, I don't interfere with that at all. I don't ask any questions, because there have been people that have gotten in trouble when you ask about giving sedation and doing all these things. I mean, there were people that brought up murder charges for that. So I don't interfere with that, but I think as a field, we are going to have to police that, and we're going to have to come to some kind of unified understanding about kind of what constitutes a dead donor. We are going to have to redefine that act.
A quick, slightly humorous comment. You mentioned that Joe Murray won the Nobel Prize. When he was here, he's the first John Nigerian lecturer, Joe Murray. He gave a nice lecture at the end of his talk. He said, my grandchildren bring up some interesting things, and his grandson said, well, Grandpa, it's true. You did win the Nobel Prize, didn't you? He said, absolutely not. And he's like, I've always thought for sure you won the Nobel Prize. He says, I did not win the prize. I was awarded the prize for the work that I did. And then his granddaughter said, well, Grandpa, I have a question too. They lived on a farm in Texas. And said, did Mom have to eat placenta after we were born? So children do bring up these ideas.
He's an interesting guy. He was a plastic surgeon.
Yes.
Yeah, his initial training, and Tom Starzl was a thoracic surgeon, and was not the conventional term of and his PhD was in neuroscience. So a lot of times you find your way, and that's a lesson too, is that oftentimes when you're early in your career, you're doing research, you're going to do one thing, and then by the time you're practicing, you're going to do something entirely different. You think you're going to go in one way, and then you're going to come out entirely different as the field changes and your ideas change.
Fantastic.
Thanks.
I'm curious. So when you're talking about xenotransplantation or organ transplant from like elderly individuals, how do you overcome the cellular senescence that varies from species to species? And
just turned to Grant about that last week, so I have a lot of answers to that. Cellular senescence is scary a little bit, because there was an interesting paper in Age18 where they took mice, and they took mice from organs from old mice, put them into young mice, and they found that the mice actually demonstrated a lot of features of the older mice, and their physiologic scores and everything dropped, that the organ can actually make the recipient senescent. And so there's ways to overcome that. And a lot of the omics studies that we're doing, and CD38 the greatest manifestation in that, and what I didn't really talk about, is it is really a driver of aging in senescence. And so we are showing now with senescent cells that targeting this type 3 CD38 will actually arrest what we call the senescence associated secretory phenotype. Senescent cells send out this inflammatory milieu that kind of affects the other cells around them and makes other cells senescent. It's almost like a cancer. It's fascinating. And so turning off that senescent signal is going to be key, because the donors are not getting younger. We're not going back to the 20-year-old donor. I mean, my phone is just exploding with offers, 75, 80, 85. And then I had a partner call me on a 90-year-old donor. And I said, come on, guys. We can wait. But I think you're right. That is a significant problem for organ transplantation. And we will be able to, once we identify the signals of senescence, be able to gene edit those out. I do think, and there's an interesting study, and you've heard about in the dog world, what's happening with dogs, is that they now there's a difference in aging between young dogs and old dogs, right? And so not young dogs, large dogs and small dogs. So large dogs die very quickly, right? They live half the lifespan of your small little dog. And so there's a drug out on the market right now that is showing, at least in the early kind of clinical data, that it will double the lifespan of large dogs by modifying the senescence signal. Now, that has not been translated to humans as of yet, but the first anti-aging drugs are coming out. And I think in your lifetime, it's going to be something that's treating. I think we will be treating with anti-aging drugs once we have a better understanding of the immunome and the genome and transcriptome and proteome.
When you originally mentioned about like all the different kind of perfusion devices that are used for the organs and the costs that are associated with them, a lot of these now, at least from what you kind of are presenting, are done by a lot of these private, like pharmaceutical companies. Mm-hmm. So is there anything that's being done to, like, what is the incentive for them to go deeper? Is it that they can do more, or do you actually feel like they're being incentivized to make it more cost effective, or is it in their best interest to do so? Like, what do you think needs to be done for that to progress?
So there's a number of things that are going to be done, and it's the neat thing about economics is self-regulating system to some degree. I think a lot of people got very enthusiastic about Transmedics and some of these other companies. They adopted the technology like crazy, and Transmedics will wipe out the margin. I mean, at 120,000, I don't know how people pay for it. I still don't know how people pay for it. But even though you'll do more transplants, there's no net benefit to doing it. And so what's going to start to happen is as people start to look at kind of how value is spent in the healthcare dollar, there's going to be some downward pressure to make it cheaper. And then so what people will do is they'll say, well, you know what, I'll just do NRP, which costs a couple $1,000s, and hook people up, and then just skip the NMP and I'll get reasonable results. And then people will stop using the devices. And so then those companies are going to have a decision to make. And the decision is going to be, am I going to make this affordable and not charge 10,000 times what it costs because it's just a bunch of plastic tubing and in a pump head. I mean, it's not in a membrane oxygenator. It's not $130,000, right? It just isn't. And so at some point, there's going to have to be some economic thought about what this looks like. And then what will also probably happen is some of the larger pharmaceutical companies may jump in and they may say, hey, we're willing to accept a larger margin. We'll buy your device and then we'll just sell it, use economy of scale, drive it lower, and so that they're willing to accept a lower margin, but do more pump cases. I mean, these things will have to self-regulate at some point. What that looks like, I don't know. But I think that this kind of expansion, and I know if you look in the news in Arizona, I think the politicians were getting involved because they were and Mayo Clinic was doing, I think, almost 100% of the organs on the pump. And so then people are starting to look at that like, whoa, what's going on here? And so I think that there's going to be some economic pressure. They're great devices. They work well. I mean, nobody can deny the effectiveness of these devices, but we're going to have some kind of common sense when it comes to kind of the cost. It's got to be a little cheaper because it can't bankrupt the system.
Not to torture people at the end of the thing, but I got to have you speculate on the science. What is you mentioned this briefly, but I want to know your opinion. What do we think the true mechanism of improvement is with machine perfusion? You talked about the things, getting rid of evil humors by just flushing, restoring ATP levels. Why does it work better than we might otherwise think it?
So would you mean HOPE or would you mean NMP?
Either one. So hope might be more washing out the evil humors, and
Yeah.
E may be ATP. But what do you think the true benefit of all these?
So I think as far as hope is concerned, I do think that it is the evil humors. I don't think it oxygenates anything. I've talked to Jim Guerrero about this. We've been in an airport chatting about this a while back, and it's hard to scientifically understand hope other than washing out the evil humor. So we'll put that aside, as I think that's how that works. And we see kind of an analog for that in the kidney world, right, where they've been doing kind of the non-oxygenated cold perfusion, and it does help in delayed graft function and things like that. I think the normothermic machine perfusion, it's interesting. I do think that I think that somehow allowing the cells to regenerate ATP and kind of not go immediately in an ice bath with the transmedics model seems to stabilize them a little bit. And, they undergo stress. And I think there's some upregulation of stress and survival systems and things like that better position it to do better over time. I think that's one thing. I think I have seen a difference between the back to base models where you allow cold ischemia versus the transmedics model where you put the organ directly on the pump and don't let it see a lot of cold ischemia. I think that the back to base model has shown, I think most people would say now it's probably somewhat inferior results to the transmedics model. And so I really think it's probably just allowing the organ to kind of metabolically kind of take a breath, to recover its ATP, it stabilizes the energetic state of the cell. I think it helps mitochondrial respiration. Throwing it in ice directly, especially when it's marginal, is probably really bad for a lot of these organs. I think that the DCD equation, and now we're having to extend our criteria, we're starting to find out what the limits of static cold storage are, and I think that normothermic perfusion allows some cellular recovery. I think the active repair, giving it therapeutics to improve its function, if you ask me, that's the real benefit of normothermic perfusion, because I think we're going to get to a point where we have to actively treat a lot of these organs. If you have an organ that's got 2 hours of ischemia, you're going to do something. I mean, that's not going to work, right? I mean, and it's 75-year-old, it's not going to work. So I think that's the true, if you ask me what the true benefit is, it isn't preservation, it's a platform to allow for assessment and repair. But a long answer to a short question, I don't know.
That was Dr. Herring's idea with islets,
Yeah.
he wanted them in culture for 2 days so that he could do a lot of interventions to repair those islets before transplant.
One thing I've clinically noted, it's really interesting, is that I think that organs do better on the pump when they've had a period of time. Like if you put them on the pump for a short period of time and then just do the transplant, I've seen less than desirable results in allowing 6 to hours or so on the pump, letting it kind of sit there and recover. They seem to do a little bit better with our back to base model. I've noticed that. It's anecdotal.
Sylvester, great talk. Thanks. I just wanted to let all the residents know that there are some unmeasurable benefits of the pump. You can put the pump, sleep overnight, and start the case in the morning. So that's something that's not quantifiable.
It'll help your life. Yeah, it's not. It's been a game changer.
The surgery has certainly become very popular because of that reason. I think more and more residents all of you can come into transplantation and not have the stressful life that most of the older people.
But I think even we're understating it. I think logistics are huge. I mean, I think that it allows us to do some interesting things. So if I get organ offers and they're coming in at the Wild West and there are like 5 of them, and I can we have multiple machines now, and it's not unusual for us to have 2 or 3 organs on the pump. And we'll sit there, and it allows us to really kind of deal with complicated logistics. Unfortunately, the OPOs don't cooperate. They'll throw an organ at you and say, hey, it's your problem, not ours. This is when it's happening. This is when you're going to do it. So it allows us to kind of stack it. It used to be tough when you get those organs and, you'd have to figure out, you do transplant after transplant. Some of my organs had 10 hours of ischemia. I mean, it's a real mess. And I think that sometimes a lot of that inefficiency of the health system got transferred risk to the recipient. I mean, when you're doing a transplant in the middle of the night, I don't know, but it gets harder the older you get, right? Like the 2 and 3, and so it's nice to start at 7. You're fresh, the team's fresh. It's just a better operation and a better experience for the patient. So that's one of the big benefits is the logistics.
So this is a question for you on your CD 38. Have you narrowed down to any drugs that you're currently working on? And are you planning to do that ex vivo on the pump or in the patient?
So we have 26 drugs that we've identified, and we've taken 2 or 3 of the most the ones that seem to work the best in animals. And so then we have another project that we're working on is that we're going to take discarded human organs that are on the pump that nobody uses, right? So for some reason, the lactate clearance isn't good enough. They're not performing on the pump. We're going to give those drugs, and then we're going to see if we can shift those back to viability. And so we're going to see what it's kind of our rescue trial. The issue with that is that it's incredibly expensive to synthesize these drugs in large amounts. And so if you think about, kind of a human organ, we're anticipating once we kind of really narrow it down to one drug that we want to start with, it's going to cost us at least $3 to $5 million to get clinical grade stuff that's produced in the right laboratory and to build up enough of the supply and stuff like that. So that's the problem. We can do it in mice all day long, but to kind of make that jump to patients is going to require a large amount of funding. But yes, there's actually, from the drug that I showed you there, we have one that's 26-fold better than 78C. Now, what's interesting though is when you take those drugs, you learn a lot about drugs by hanging out with medicinal chemists. But there's also, the drug can work great as an inhibitor, but then the body's pharmacokinetics and pharmacodynamics are difficult. The absorption, there's lots of stuff you have to deal with. And so even that drug has to be re-engineered. And ideally, we had to add side chains and change the chemistry. So there's a lot to work on. And so we're always kind of trying to get grants and money as much as we can. And it works in multiple organs. It's not just liver. It works in the lung. It works in the heart. We have a heart study where we ligate coronary arteries, and we're able to shrink the ischemic area of these with CD38 inhibitors significantly.
I have a comment from the chat from
Yes.
Dr. James Lee.
Yes.
A terrific lecture that will be seen as a high watermark in the department's history. Thanks.
Oh, wow. Thank you.
Sly, that was a fantastic talk. It was really, for me, nice to hear about the evolution of your time here in Minnesota and how that shaped your career and all the great things that you're doing. I think along Eric's lines, that really looking forward to seeing you implement some of these into the kind of clinical use. The use of pumps is kind of ubiquitous. I think the next frontier, as you mentioned, is really intervention with pumps. That's going to make a lot of difference.
Absolutely. Thank you.